Stanford University.
Now I'd like to start this evening by introducing someone who couldn't be here with us last week and who is the co-director for this course, and that's Dr. Sherry Wren, who's right here. Dr. Wren is well-deserving of your applause. She is currently a professor of surgery, an expert in oncologic surgery and chief of surgery at the VA hospital, of course, which is affiliated with Stanford Medical School. But beyond that, she is a person of really quite significant renown in education. She's won virtually every teaching award that we've given. She's served as the director of the Medical Senate, which is really quite an esteemed role. And she's also nationally recognized for her work in surgery as a governor of the American College of Surgeons, and this year became an associate dean in academic affairs. So we're really pleased to have Sherry with us for the course. Thank you, Sherry. And because this course has become so popular, our speaker for next week, Jill Helms. Jill, why don't you stand up? She couldn't wait to be here. In fact, she asked whether she could go first tonight, but I told her no, she'd have to wait till next week because we're going to drill down tonight even to smaller entities than what she's going to speak about next week. She'll talk about the fascinating area of stem cell biology and regenerative medicine, but tonight we're going right down to the molecular basis of life. And this is a really important topic, one that has certainly changed, as I indicated to you last week, in just such remarkable ways over the last 40 to 50 years, really with some of the most fundamental work taking place here at Stanford. Just to remind you of what I said last week, that began when the school was really founded here in 1959 with Arthur Kornberg's seminal investigations. He won the Nobel Prize for his work, and just 2 years ago, his son, Roger Kornberg, also won the Nobel Prize to kind of bookend that family and really catalog some of the extraordinary work in the study of DNA and its transcription. As we think about the future of our topics, tonight's session will be really, I think, quite essential for your knowledge base. Now, our speaker tonight, Professor Gil Chu, has an interesting history in his entree into medicine. He started out at Princeton University, where he was a extraordinary student in physics, actually went on and got his PhD in physics at MIT, and then a light went off and he said, I think I'd like to be a doctor. And so he then went to Harvard Medical School, where he completed his MD degree, trained in internal medicine at the Mass General, one of the most distinguished hospitals in the nation, and then elected to move into the area of oncology, came to Stanford to do his fellowship in medical oncology, joined the faculty, and has had a remarkably distinguished career since then, both as a professor of medicine and as a professor of biochemistry. He works in the area of DNA repair, damage related to both ionized radiation, ultraviolet light, which may sound ethereal, but is actually related to some disorders that fall into my own world of pediatric oncology, where risks for UV light damage can actually lead to certain malignancies. So he is someone who's leading the edge, if you will, in fundamental new knowledge, and beyond that has also been one of our most distinguished teachers. He is the person who leads the learning curve of our new medical students in the foundations of molecular medicine. So I'm really pleased to have this evening for you and with you, Dr. Gil Chu.
So thank you, Phil, for that very nice introduction. We have a lot to cover. The 3 R's we're going to do tonight. Usually that takes 6 years in elementary school, and I'm going to try to do it in less than an hour so that I can get into applications of the 3 R's, because you don't want to just learn about the 3 R's. And we're going to go all the way from molecules to medicine. This is in Braun Auditorium. That's the periodic table, and I've got my molecules here and my periodic table here. So if you forget, I'm wearing the tie. So the way this talk is organized, and I don't think there's a laser pointer, but I may not really need it, but it's okay.
Let's hope not.
3 R's are replication, recombination, and repair, and I'll tell you about them. And then I'll tell you about a couple of very short applications using recombinant DNA and new drugs. And then implications. Implications of the 3 R's, the application of the technology to new drugs, and implications that has for our healthcare system, which you've probably heard a lot about lately. So we're going to go right into the 3 R's. Replication copies DNA, recombination rearranges DNA, and repair fixes DNA. And I'm going to start talking about some complicated things, but I just wanted you to know what replication does is it, like on the Starship Enterprise, that replicated food, and what replication does is it makes an exact copy of DNA, just as on the Starship Enterprise. Now, when we start getting more complicated, don't hesitate to raise your hand and interrupt me, because if I've skipped a step, I don't want to keep moving on. I want you to interrupt me and ask a question, okay? If you have a philosophical question, we can wait till the end. All right. Now, Phil mentioned Arthur Kornberg, so here's a picture of Arthur. He actually founded one of my 2 departments, the biochemistry department, by bringing it lock, stock, and barrel from St. Louis. And in 1957, he was able to reconstitute the process of DNA replication in a test tube. The headlines at the time said, Professor reproduces life in the test tube. Not quite right, but he reproduced a really key step in life. And he found that the reaction only required a few components, which I've listed here in red. A DNA polymerase, which copies the DNA, a template DNA, which is the DNA to be copied, primer DNA, to get the DNA polymerase started just the way you prime a pump. That's why it's called primer DNA. And nucleotide bases, which are incorporated into the DNA. And you all know that there are 4 bases, which are basically the code for life. And how they're organized in the DNA instructs the cell on what proteins to make and how to make them. All right. So basically, what you have is you start out with a DNA molecule, and if the cell wants to divide, it has to make a new copy. And this is an electron microscopic image of the replication of the E. coli genome. E. coli, there are more genomes of E. coli in you than your own genome, because there are a gazillion E. coli sitting in your gut. And so they're very handy. We don't have to dig very far to find them. And that DNA is being replicated, and these things are replication forks, which are marked by those arrows. And those are the areas in which new DNA is being synthesized. And those forks are moving outward until you finally end up with 2 circles, and then the 2 circles will go into 2 daughter E. coli bacteria when it divides. Okay, same thing with your cells. So here's where it gets a little complicated, but I need to give you a little bit of a picture because I have to show you the replication fork in order to show you some of the applications later. So replication differs. There are 2 strands of DNA. Okay, it forms a double helix. And the DNA actually has a directionality to it, and we call it 5' to 3'. So you'll notice that the DNA molecules are anti-parallel to each other. They each have a 5' to 3' direction, but the 2 DNA strands are going in opposite directions. So the 5' and 3' ends of the 2 molecules are next to each other up there. All right? Now, it turns out DNA polymerase is kind of dumb. It only goes in one direction, 5' to 3'. It doesn't go backwards. There are actually very deep reasons why it doesn't go backwards. But it's very important for it not to go backwards. Okay? Now, since the 2 DNA strands are anti-parallel, if you're going to copy that DNA at a replication fork, One of the strands is very easy because on the upper strand, you're copying the yellow DNA into new red DNA, and because you're copying it in an anti-parallel direction, the DNA that's being synthesized is going 5' to 3' opposite to the template DNA strand, the old DNA strand. So that polymerase keeps going in a continuous way and then displaces the replication fork to the left. On the other hand, so from the left-hand picture to the right-hand picture, you can see the replication fork moving to the left. My left, your right. Okay? Going that way. All right. Now, because the replication fork is going that way, and because the lagging strand, the other strand of DNA, is in the anti-parallel direction, and because DNA polymerase can only go 5' to 3', it can only go backwards away from the replication fork. So what it does is it goes backwards for a little bit, and then it hops towards the replication fork and goes backwards again, creating very short fragments, which were discovered by a molecular biologist in Japan named Okazaki. So they're called short Okazaki fragments. And because it's going backwards, each of those Okazaki fragments can then be synthesized to meet. So on the lagging strand, an Okazaki fragment gets synthesized until it bumps into the previous Okazaki fragment, and then a ligase, a DNA ligase, joins the two DNA ends. Are you with me so far? Uh-huh. Okay, so, yeah. Go ahead.
So, if it has two sides, a 5' and a 3', if it's taking one that's going from 3' to 5', making one that's 5' to 3', it's making it backwards.
That's right.
How is it replicating things it hasn't gotten to yet?
It's not. It's only going to get to, so on the top strand, that one's easy, right? That's going 5' to 3', and you notice that the new DNA that's being synthesized will therefore be anti-parallel to the DNA it's copying. So the red strand on the upper fork is going in the opposite direction from the yellow strand on the upper fork. And that's going to keep going, displacing the fork. So the fork is going to get displaced and unwound.
It's the same basis, but facing the other way?
Okay, so the question is it the same basis but facing the other way? It's, quote, copying the DNA, but it's not copying it as an exact copy. What it's doing is, it's every time it sees the base A, it puts in a T. Every time it sees the base G, it puts in a C. So it puts in what's called a complementary base. A's and T's are complementary to each other. G's and C's are complementary to each other. So the code is actually got a code of G, C, A, T, but the other strand has a code that's kind of the bizarro mirror image of it. It's not the exact image, but it's the complementary image. Okay? Did you ever read Superman comics? So Superman was, the knight for all good, but there was a bizarro world, which was a copy of Superman's world, and there was a bizarro Superman who kind of looked like Superman but wasn't quite the same. That's what this is. You're putting in complementary bases of DNA. So since you're always putting in the complementary bases of DNA, when you copy that other strand again, you just put in the complement to the complement, which restores the old strand. So as long as you're putting in the complement each time, you've always got all the information there. Ooh, thank you. Are you okay with that?
Yeah,
The Superman thing worked. Okay. Uh-huh.
Is there just one polymerase molecule that's taking care of both these strands, or is there like a sea of these things floating around?
No. Actually, there's a big machine called the replosome, and actually, how many people here sew? Yeah, mostly women. I'm looking. Not a whole lot of men are raising their hands. But the way a sewing machine works is this strand is working like a sewing machine. So it's going backwards, and then it goes this way, and then goes this way, right? Just like a sewing machine. So there's a polymerase that's doing this job, and there's another polymerase that's doing that job. But they're all part of a big machine. So the whole thing is being held together right at this fork, and then this fork is moving. Is that okay? Okay, yeah. I forgot to repeat the question, but let me try to repeat.
The fragments that it works in?
Oh, what's the length of the fragments in which it works?
Is it standard, or does it do it in like 10-bit pieces? Or?
Yeah. No, this leading strand, the question was how long does it do the replication? Yeah. On the leading strand, on the lagging strand. Okay, so I'll start on the leading strand. The leading strand is really long. It just goes and goes and goes. Okay? In fact, there's a sliding clamp that keeps it on. And the sliding clamp looks like a clamp. It's like a circle. On the lagging strand, what it'll do is depending on what organism you are, if you're a bacterium or a human, it's slightly different. So it's a few hundred to many hundreds of bases. So like a sewing machine, the length of the stitch depends on the species, but it's fairly short, a few hundred. Just to give you an idea, the human genome has 6 billion base pairs. So it's just little snippets. We're okay?
So at what point, when it, I guess, reconstitute itself, how long would it be before it actually becomes just like...
2 molecules, yeah. So in E. coli, in the bacteria in our guts, they can divide in about 20 minutes. Takes us a little longer. It takes us a little longer. So the question was, how long does this process take? And an E. coli can do it. Their DNA genome is only 5 million base pairs. They start at one spot and they go. We've got 23 chromosomes, so we obviously, or 46, but every chromosome starts independently. But because our genome is so large, we actually initiate replication at many spots on each chromosome. So we can get the job done in a reasonable amount of time. Okay, so let's move on. That was pretty tough, actually. I'm hoping that's the toughest. Okay. Now, I've tried to make it simple. We're moving on to recombination. That rearranges the DNA. So those of you who don't know what rearrangement is, you remember Mr. Potato Head. And this is Mr. Potato Head Optimash Prime. Do you watch those kind of movies? My teenage son does. But anyway, Transformers. Okay. So anyway, this is now Optimash Prime. You've got a mustache here. I don't know what this is. Some kind of a nose, a different thing out for the arms. I don't know. Maybe a beard. I don't know. But you can make all kinds of Mr. Potato Heads. And DNA recombination is a way of rearranging the DNA to make new pieces. Okay? It turns out that kind of stuff is going on all the time. You probably thought that you were born with a fixed complement of DNA and your future was set. But I want to disabuse you of that notion. You are actually being bombarded by changes all the time. Your DNA is constantly in flux by this Mr. Potato Head-like system. So you need to hear about recombination. Okay? Now, there are 2 types of recombination. And one is called homologous recombination. No, this is going to be a hard one. Homologous recombination. But we'll get you through it. And the other one is site-specific recombination. So here goes. Watch homologous recombination. It's a mess. First of all, I have to tell you what the word homologous means. DNA molecules are homologous if they have similar DNA sequences. 2 identical twins, when they're born, before mutations occur and before all kinds of recombination occurs, are 100% homologous. That's what an identical twin is. Certainly, in utero, they're 100% homologous. Now, men and women are 97% homologous. Chimps and humans, male chimp and a male human, are 97% homologous. So, that explains a lot. But I think you got the concept. They're pretty similar, but there's some important differences. Okay. All right, now, this is the hard thing. What a mess that is. homologous recombination repairs DNA double-strand breaks. So I started out with 2 DNA molecules that were intact. One was a double-stranded DNA molecule that was yellow, and one was a double-stranded DNA molecule that was red. Okay? Now, the yellow molecule developed a double-strand break. Now, a double-strand break is a disaster because that's a broken chromosome, and the cell will die unless it can fix it. So what homologous recombination does is the yellow DNA, double-stranded DNA molecule, was the broken one, right? And so what it does is, when it's broken, what often happens is some of the stuff at the ends gets all screwed up, okay? And in fact, the information gets messed up. So what it does is it says, aha, if I can find a nearby DNA molecule that's homologous, I will use that to do my repair. Okay? So what it does is, it does something called strand invasion. It takes the 3', remember I told you about 5' and 3' ends? Ooh. It takes the 3' end of this DNA molecule and it invades the homologous DNA. Now remember, the DNA molecules, the 2 DNA strands were complementary to each other, so they will actually hybridize to each other by hydrogen bonding. Okay? Hybridize means that the 2 DNA molecules will stick together because it turns out that there are nice hydrogen bonds between the G nucleotide and the C nucleotide that cause them to hydrogen bond and stick together. And there are hydrogen bonds between A and T nucleotides that cause them to stick. So the DNA molecules will actually zip up because the complementary bases want to stick together via hydrogen bonds. That guy. I can't teach you all of chemistry, but I think you remember some of that. Okay? These are not covalent bonds, but they are bonds that nevertheless will cause 2 molecules to stick together. So each double-stranded DNA molecule wants to stick to itself, but under the conditions of homologous recombination, you displace one of the strands to allow this broken DNA molecule to strand invade. Once it strands invades, then DNA polymerase, remember that guy, can actually copy this DNA. So if the double-strand break was created where you lost a few bases at the ends, okay, you can recover them by copying the missing bases off of the other DNA strand. Now, I'm not real good at fixing things unless I have a copy to work from. So when I start doing plumbing and I forgot how I messed things up, I go to the other bathroom and I check it out and then I copy what happened. I've got a bathroom in the house with 2 sinks right next to each other. That's very handy. So this is what's going on here. Okay? Then, after you do this, the other side of the break actually can copy off of the displaced strand. All right? Then what you can do is you can hop back and you get something called a double holiday junction, which you'll notice has 2 L's in it, so it's not a vacation. It's named after a British biologist, Robin Holliday. And what happens is these junctions actually get resolved by nicking the DNA in either one of 2 ways, and the DNA strands are cut at the arrows, and then you end up with 2 intact chromosomes. Chromosomes with something called crossover and something without. Now, you don't have to remember that for later. But you end up now, depending on how you cut the DNA, it doesn't really matter, you end up with 2 intact chromosomes. Now, some information, quote, has been exchanged. So if you're copying a DNA molecule that's 99% the same, you'll copy the mistakes, the 1% of the mistakes. Okay, you okay with this? I think this is the second hardest slide. Questions?
Did we already have the first hardest?
Yeah, you did. I think getting started is the hardest.
What causes the strand break in the first place?
What causes the strand break in the first place is the question. Just being alive. All the time. You're making oxygen-free radicals while you're metabolizing. You're at body temperature right now, and oxygen-free radicals are coming in, and they are making strand breaks in your DNA. And when they make 2 strand breaks that are near each other, your DNA falls apart. The other thing is cosmic rays are coming in and getting you. Okay? Or you went to the dentist. I went to the dentist yesterday and he wanted 14 x-rays. Bang! X-rays are especially good at making double-strand breaks. So just being around causes this to happen. So this better be working or you're in trouble. Uh-huh.
Is it only ionizing radiation that does that? Can RF energy create strand
No,
breaks?
well, in terms of the electromagnetic spectrum, you actually need enough energy, high enough energy, and tuned enough to break the strands of DNA. Ultraviolet radiation, for example, will not do it. Ionizing radiation is tuned just right to break it. But there are other things that do it. Plants, for example, make a lot of poisons, and they have drugs that will actually create double-strand breaks. In oncology, we harvest those drugs and give them to our patients, not because we don't like our patients, but because they happen to kill cancer cells just a little bit before they kill the patient. And so they have some benefit for cancer chemotherapy. And some of our most important cancer chemotherapy drugs make double-strand breaks. So lots of ways of making double-strand breaks. It turns out your immune system, in order to generate immunological diversity, makes double-strand breaks on purpose to generate huge numbers of different kinds of antibodies. So this is just happening.
Where's this spare DNA come from that it's grabbing onto? Is that
Where's the spare DNA come from? This process only operates during replication. Where did the spare DNA come from? All I have to do is go back to the replication fork, because we spent so much time on this. If there's a double-strand break over here, there's the spare DNA, right on the other side of the replication fork. Now you're going to ask me another question. What's the other question? Remember, this is always happening. When replication ain't going on. Right, when replication isn't going on, there is another machinery that actually repairs the breaks, which I'm not going to tell you about. It happens to be what I dearly love, because it's one of the things I spend all my time thinking about in the laboratory. But I'll spare you the pain. It's actually quite simple. It just jams the DNA ends together. And if it doesn't work, you don't have an immune system. So great question. Okay. Any other questions? I think most of you are coming along. Do you understand the questions? Okay, yeah.
No, what assures that the proper DNA is used to make the repair?
Yeah. Well, what happens is this only happens during the replication, right? And the strand invasion only occurs between homologous DNA strands.
So what percentage homology do you have to have?
Ooh, what percentage homology do you have to have? It's got to be at least 95% or so. Otherwise, it's shades of gray. If it's 100% homology, the strand invasion is very efficient. If it's 98%, it's pretty efficient. I think, somewhere around 90%, it just doesn't happen very often. But you're asking a really good question, because maybe there's some place else on this newly replicated DNA that it could hop over to. And sometimes it does. And when it does, you could have a deletion. So mistakes happen. Mistakes happen. That's why he's worried about errors. He's asking, how homologous do they have to be? Right. Very good. Now, replication occurs with very high fidelity. 1 error in 10 to the 7 bases. So if you do homologous recombination right here at the replication fork, very few errors. But it turns out that homologous recombination also operates during meiosis, when you're making egg and sperm. And there it occurs between different chromosomes. And there's where we get all the diversity I see in front of me. Although, I don't think you're as diverse as our medical school class. No, you're definitely not. But we'll work on that. Okay. But this thing can operate when it's not precise. And there's a special machinery for making it work during meiosis. Okay. So we're good with this. Now, yes?
With the replication machine, when it has an A and it needs to make a C, is it on the other end?
It happens to be a T, but it's not important. Yeah.
Does it create a T, or does it find a T?
Yeah. So the question was, what about this replication machine? It's coming along and it sees an A and it needs to copy the A. And the A is going to be copied into a T. But remember what Arthur Kornberg did. He found out that you have to have A's, T's, G's, and C's in the reaction. So those things are just floating around in the cell. And so when it hits an A at this point, it's going to stick in a T there because they're floating around. There are T's floating around. Is that okay? Yeah. All 4 nucleotides have to be there. So it'll just grab one and bring it in. So that part was easy for it to do. It doesn't have to search. Okay, good. Aha.
You mentioned an A or a T. There's no indication on the graph where you wonder, what about the C and the G? Are they?
Yeah. So just imagine, there's some sequence here. And the sequence could be G, C, A, T, whatever. And they're all in there, yeah. And then when it hits a G, when it sees a G here, it goes, I have to put in a C. And then, I didn't repeat the question. He says, did you get the question from my answer? So he's saying, what's going on here, basically? What's in this sequence? I don't see a G, C, A, and T. That's because I left it out. But there are G, C, A, T, G, G, T, 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 whatever. Okay, and when the polymerase hits a G here, it'll put a C opposite, and then it'll move a little bit forward and stick in another nucleotide and so on. Great. Aha.
What is the speed at which the breaking and joining is happening? And does the speed change over age?
The speed of the repair or the
breakup and the repair.
How fast does this repair occur? I don't know. Somebody may know. I don't. That's not a well-defined number. I know for the pathway that I'm interested in, non-homologous end joining, it's over the span of hours. So, for homologous recombination, my guess is it's probably of that order. So it takes the question was, how long does it take to do this repair? It takes a couple hours sometimes. And so there have to be all kinds of signals to tell a cell, hold it, don't do anything yet. I've got to fix things. Okay?
How long is for the breakup? Say 2 hours?
How long does it take to make a break?
Yeah.
Well, how long does it take to break something? They know the answer. Cosmic ray, bang, it's done. Right? So it's always easy to break things. Yeah. So I think your real question was, how often do the breaks occur in my body? That's your real question. What's the frequency of breaks? It's happening all the time. Yeah. It's happening all the time. I'll get at some really hard numbers. It'll really scare you. Uh-huh.
Is the repair as vital in a young person as it is in an older person?
Is the repair as vital in a young person as it is in an old person? Well, I'm older, so it's more vital to me. But I think it's equally vital, really. It turns out that if you don't repair things properly, mutations occur. And if you build up enough mutations, you get cancer. And on average, you have to mutate about 6 very important genes in order to get cancer. And as you age, you sort of build, each one of your cells builds up a few mutations. And if one of them happens to get the magical 6, it can turn into a cancer cell. So if you look at the incidence of cancer as a function of time, it goes up as the 6th power of age. Whoa. T to the 6th power. That's why old people get cancer and young people don't. But the cancers that young people get are the cancers that only need one hit. So there are a few rare cancers that just need that one hit. But most of the common ones, breast, colon, prostate cancer, pancreatic cancer, those need, on average, about 6 hits. Uh-huh.
Related to the other question, how long can it be broken for this mechanism to still work?
How long can the DNA be broken for this mechanism to work? Well, if the DNA is broken and the replication fork gets too far away, then you have to pull in the backup pathway, the non-homologous end joining pathway. That it's non-homologous because it isn't homologous recombination. So we call it non-homologous end joining.
But the repair will start nonetheless.
It'll try to do it. Yeah, it'll see the break. All kinds of signals will go out. Ah, broken, right? And then it'll try to bring in all the machinery to repair the break. Lucky for you. Yeah. Now, here's some more machinery. This is going to be important later. Several proteins promote strand exchange. Remember I told you that in order to have this strand exchange, you have to take a DNA molecule that's already zipped up and somehow unzip it to let that broken piece of DNA squeeze in. Right? Remember that? So what happens is that broken DNA actually, this is one of those things where you take a few steps back in order to go forwards. The broken DNA actually gets resected, leaving a 3' overhang. You actually remove some pretty good material. And then that leaves a single-stranded piece of DNA. And that's a single-stranded binding protein called RPA, replication protein A. And that coats the single-stranded DNA. That's really to protect it temporarily. So that just coats it to protect it. And then two proteins, RAD51 and BRCA2. BRCA2 might have a familiar ring to it because BRCA2 stands for breast cancer 2 gene. So we're very interested in that. RAD51 stands for radiation because it was first discovered in yeast, and yeast that were mutated in that gene were especially sensitive to ionizing radiation, which makes double-strand breaks. So that's why it's named RAD51. Now those two proteins make it to this junction between double-stranded and single-stranded DNA. And what they do is they see this protected piece of single-stranded DNA, and they load on and they cooperate so that more and more RAD51 molecules get recruited to make a filament on this single-stranded DNA. And it is that filament that actually makes this especially good at doing strand invasion to create what we call this D loop. It's called the D loop because biologists, okay, are really cockeyed, and sometimes they walk around like that, and there's a D there, okay? So these filaments allow that D loop to form, and then, because that's a reaction that doesn't want to happen all by itself, okay? And then, now you can copy and repair your break and so on, and then you're free and clear, all right? Now, it turns out that BRCA2 is loading RAD51 on, okay? So it's just ushering the RAD51 onto the single-stranded DNA. But BRCA2 has to have some help to get where it needs to go, and guess what? BRCA1 helps BRCA2 get there. BRCA1 has a familiar ring. It's also involved in breast cancer. Okay, and BRCA1 guides BRCA2 to the DNA end. Some of this we just discovered last year. But it's so neat, you have to hear about it. Because it all makes sense now, right? Okay. Not me, but they discovered last year. Okay. Now, I want to show you a filament, because I promised you molecules to medicine. Here are the filaments. This is what it looks like. The RAD51 molecules are wrapped around the DNA, and now you can see the wrapping. Can you see the wrapping with it? You know, the lights are shining on me, but I think you should see the wrapping, which looks like that, wrapping around the DNA. So that's the active ingredient that causes single-strand invasion. All right. Now, BRCA1 and BRCA2 mutations cause breast and ovarian cancer. Affected individuals carry one mutant and one normal allele for BRCA1 or BRCA2. So one of their two chromosomes has a mutation, the other one doesn't. But if you get a mutation of the second allele in a breast cell, it disrupts homologous recombination because you can't load RAD51 on anymore, and you can't do the strand invasion. So homologous recombination gets shut down. Now, because you don't do homologous recombination, you don't repair double-strand breaks as efficiently as you once did, and as a result, you develop more and more mutations. And it's it's easier to get those 6 mutations that you need to become a cancer cell because you have a repair system that's been knocked out. So women who carry these mutations will get breast cancer in their 20s instead of in their 50s and 60s. It takes much less time because they've knocked out this repair machinery. You okay with this? Yeah. Allele. What does the word allele mean? I should kick myself. That, I apologize. Allele just means that I have 2 chromosome 6s. Chromosome 6, chromosome 6. Therefore, I forget which chromosome BRCA1 is on, but there's one gene on this chromosome, BRCA1 gene on this chromosome, and one BRCA1 gene on this chromosome. Okay? Those are the 2 alleles. So allele is a fancy word for saying I've got 2 copies of every gene, with a few exceptions, right? We men lack 2 copies of some very important genes. Women, on the other hand, this may be an explanation for their superiority, have 2 copies because they have 2 X chromosomes, right? We only have 1 copy. But except for the X and Y chromosomes, we have 2 copies of every gene. The unfortunate nature of the fact that we have only 1 X chromosome means we're susceptible to a lot of diseases because we don't have a backup copy. So that's basically the root cause of our inferiority. On the other hand, and that's why we have to subjugate women for centuries in order, but they're getting even now. So on the other hand, we have a Y chromosome, and the Y chromosome is what causes us to become men. So that encodes genes for pro football and things like that. So, those testosterone-related things. Okay, any other questions? But thank you for allele. That was very important. Uh-huh.
So if somebody gets tested for BRCA1 and 2, they already have it in their body, but they don't have the mutant if they're negative?
Oh, yeah. So what does it mean for a woman to be tested for BRCA1 or BRCA2? Right. What it means is that you're going to look at her normal cells. You really don't know if she has a breast cancer brewing, right? You're just going to take her normal cells, and you're going to see if one of the 2 alleles has a mutation in either one of those genes. Now, those genes are inherited. So that woman got one of those mutant genes from either mom or dad, and sister or brother also could be carrying 1 copy of those mutant genes. Yes. And so as a result, if you find one of those mutant genes, it tells you that the process of becoming a breast cancer has been foreshortened, and you're at very high risk for getting breast cancer. So if you turn out to be a carrier for a mutation in BRCA1 or BRCA2, you either are going to say to yourself, I have to be very careful and have very careful surveillance for breast cancer, or I might decide to have radical surgery to remove the offending organs, either the breasts and or the ovaries. So I can't get breast or ovarian cancer. So that's a very difficult choice some women have to make. Okay. Now, how do you do surveillance? This all comes around, right? How do you do surveillance for breast cancer? What do you use? A mammogram. What does a mammogram use? Ionizing radiation. What do ionizing radiation gamma rays do? They make double-strand breaks, which are repaired by the homologous recombination, which is encoded by BRCA1 and BRCA, which BRCA1 and BRCA2 are involved in. So some of the machinery, or some of the devices we have, boy, you guys have learned fast. You now realize what a problem we have. So we're sort of on Occam's razor here. Uh-huh.
So if this is the way all humans repair their cells, why does breast cancer seem to be more prevalent in women?
Oh, what a great question. So all the cells repair DNA the same way with this machinery. All of our cells are using this machinery. So how come women get stuck with breast cancer. That seems unfair. Now, that question is actually a really deep question. It almost falls into the realm of philosophical discussion, because the answer is we don't know. We do know that you can get ovarian cancer, so it's not just breast cancer, but then why? Yeah, that's just women too. Why not every other cancer? Why not? We just don't know. We don't really understand. That's all I can tell you. Now, it turns out men who have mutations in BRCA1 or BRCA2 can sometimes get breast cancer. They can't get ovarian cancer, obviously, but they can get breast cancer. So that does happen. But we really don't know. Why is disruption of this particular DNA repair pathway so important for the breast and relatively less important for other tissues? I know no one knows the answer to that. Uh-huh.
Could that be maybe why it's continuum male with a Y chromosome then, that something is actually protecting them from the breast cancer?
Yeah, the question is, are men being protected from breast cancer? Well, I think we men are being protected from breast cancer because we have a lot less at risk. We don't carry around breasts. And also breast cancers actually are estrogen dependent, and we're not making estrogen. So I think it's not got to do with the Y chromosome. It's got more to do with the fact that we have less breast tissue and we don't have estrogen hanging around that are stimulating the breast cancer cells to divide. But there's enough around so that men who have this mutation can actually get breast cancer. Breast cancer does occur in men, but it's mostly because we don't make estrogen, I think. And we treat breast cancer with anti-estrogens. So, all right. Now I'm going to tell you about site-specific recombination. Retroviral integration is one example. And I'm going to tell you that because retroviruses, oh, what's the word retrovirus mean? Ha! Retrovirus refers to the fact that these viruses don't have DNA in their genome. They have RNA in their genome. And in order to copy their RNA, they actually copy their RNA into DNA, which is kind of backwards, because usually DNA gets transcribed into RNA to make proteins. So retroviruses do it backwards, so they're called retroviruses. Okay, they cause human disease. HIV causes AIDS. You all know that. Hepatitis B, I bet you didn't know, was a retrovirus, but it actually goes through an RNA to DNA copying mechanism, and it causes hepatitis and liver cancer. And retroviruses undergo recombination with the DNA of the human genome. You may not have known that. Retroviruses are hopping into your DNA. So this is what happens. This is the retroviral DNA. It's already copied itself. It started out as an RNA molecule, but it made itself into a double-stranded DNA molecule by doing its reverse transcription. And this blue here is the target, the human chromosome. And this viral DNA, the virus carries with it an enzyme called integrase. The name integrase stands for its ability to integrate the viral DNA into our genome. So that's why it's called integrase. And it basically attacks us by sticking itself right smack into the middle of our genomes, basically at random sites. And then from there, it hijacks our machinery to make more virus particles and to move on with its life at our expense. Okay? Just an example of site-specific recombination. That is Mr. Potato Head, except it's an unattractive mustache that's being stuck on there. Okay? Uh-huh.
So do these viruses carry around their own machinery for doing that reverse transcription?
Yes. Yes, they do. It's a special DNA polymerase that instead of copying DNA, the question was, do the retroviruses carry their own special machinery to accomplish this strange maneuver? And the answer is yes. They carry the enzymes in the little virion particle.
And do we have any need for anything like that? I mean, is that a site that, say, a pharmaceutical maker might target?
And yeah, and because the retrovirus is somewhat different, he's saying, because it carries its own special enzymes, its integrase and its own special DNA polymerase, is that something that a pharmaceutical manufacturer would target? And the answer is yes. The first HIV drug that was ever developed was called azithromycin, AZT. And it is a nucleotide, a fake nucleotide, that actually fools the retroviral polymerase much more than it fools our polymerase. So that's why it worked. It didn't work. It has side effects because it's not perfect, because our polymerases also use it to some extent. But it's the other polymerase, the retroviral polymerase, likes it much more. So exactly right. If you understand the differences at the molecular level, you could be an entrepreneur, just like our friend here, who could think of a way to cure AIDS. So that's why we need to know this stuff. Okay. Finally, repair. Okay? That's what fixes DNA. We already talked about one process of repair because it's basically a recombination. It's recombinational repair. I am going to tell you that DNA repair is very important because DNA damage is unavoidable. The human genome is 6 billion base pairs, counting all the chromosomes. They're 0.34, a third of a nanometer per base pair. Now, if you multiply 6 billion times a third of a nanometer, you get 2 meters. So every cell in your body has DNA that would basically be more than most of your height. Okay? Now, your body, on the other hand, has 10 to the 13 cells. So you have to multiply 2 meters times 10 to the 13. That's 2 times 10 to the 13 meters, which means nothing to you. So I recalculated it in terms of solar system diameters. 50 solar system diameters of DNA in your little body. And damage in any one part of that can eventually lead to cancer. That's a lot to protect. So this is very important. And also you want to know, because DNA is damaged by many anti-cancer drugs, and of course, no surprise to you, many anti-cancer drugs can cause something. What do they cause? Cancer. So we take a patient with Hodgkin's disease, treat them with these wonderful drugs, and we say you're cured, and then 5 years later, they get another cancer, sometimes. At a rate of about 5%. It's usually leukemia. And so, actually, I think Phil ended up taking care of them. So I basically gave him my business 5% of the time. So that's a very unfortunate thing. We do need better things than this. Okay. So there are 3 major classes of DNA damage, and each class has many mechanisms, and I don't have time to tell you about all of them. I am only going to tell you about one. And I'm going to tell you about this one because it's going to really scare you. It's called base loss. These bases are attached to the DNA. These are the bases that hold the code. They are the G's, C's, A's, and T's. They do all the coding. Okay? Now, these things, believe it or not, fall off. About 5,000 of them fall off of the DNA in a given cell per day. So you have all this DNA. It's pretty long. It's 50 solar system diameters. So from that point of view, 5,000 isn't a whole lot. But 5,000 is a pretty high number. And so there has to be a repair system to deal with those 5,000. Okay? And I'm going to just tell you about that repair system. Because then you'll be really caught up about an article that was published last month in the New England Journal of Medicine, if you know about this. So base excision repair deals with it. See, here's a piece of DNA. Look at that. G, C, T, A, T, C, C. So this is something somewhere in your genome, right? And look at that. All the bases are little colored blocks, and there's one colored block there that's missing. DNA helix with a missing base. 5,000 of those bases fell off that one cell in one day. So that spontaneous loss of a base creates an apurinic or apyrimidinic site. That's an AP site. Now, the bases fall into 2 classes, purines or pyrimidines. So biologists came up with a fancy name, which is it should be just an A-basic site, but I'm going to call it an AP site because that's what biologists call it. Just, pretend. You can pretend I never said it, but base excision repair restores the lost bases. So there's an AP endonuclease that recognizes that AP site, the site without the base, okay, and it places a nick there. And so you have a DNA helix with a little gap. Now, if you have a DNA helix with a little gap, you know how to repair gaps because you've got a template strand to copy from, and DNA polymerase can come in and copy. And now, opposite this G goes a C. You okay? So this whole course, the whole lecture is starting to come together, right? Okay. Okay. And now you're repaired. So you're safe. Now, there is a protein called PARP. I'll tell you in a minute what PARP stands for. It binds to the DNA gap to make this process more efficient. You can imagine that you notice in BRCA1 and BRCA2, you had a protein that did the job, but then you had another protein to help it do it faster. That's because this stuff is so important. You've got to go chop, chop as fast as possible. So PARP accelerates this, and this has got to go really fast because this is happening at the rate of 5,000 per day. Now, double-strand breaks, you're only getting a few in your cell per day. But this is several thousand per day. Okay? So what's PARP? PARP recruits repair proteins to the DNA single-strand breaks, and it stands for poly ADP ribose polymerase. ADP is like the brother of ATP. That's the energy molecule. Well, it turns out ADP isn't just for energy. What it can do is it can be constructed to make big polymers of itself. So PARP actually binds to the DNA gap, and then it becomes activated, and then it attaches ADP ribose polymers to itself and to other proteins. It makes a cloud of ADP polymers. And we don't exactly know why it's doing this, but we do know that its ability to do this is very important for recruiting the DNA polymerase and ligase to the single-strand break to do the repair. So think of it as slime. These ADP polymers are just sort of hanging out there. They're very unstructured. And I think what's going on is it's like Velcro, or slime, just stuck out there, so that these very important enzymes find their way there and do the job very quickly. Okay? I mainly had to tell you about that so you knew what PARP meant, but it's kind of cool anyway. Now, homologous recombination saves PARP1 deficient cells. People were trying to figure out what PARP1 did, so they made what's called a knockout mouse. You can do genetics on a mouse and take a gene and wipe it out and say, if I wipe out both copies or both alleles of the PARP1 gene, just make it totally PARP1 deficient, what happens to the mouse? It turned out the mouse was perfectly healthy. So I didn't pay attention to the PARP literature. I said to myself, how important could it be? A mouse can live just fine without it. And then some important things started happening. I started having to learn about it. But it turns out that the reason the PARP1 mouse is perfectly happy is because homologous recombination is a backup for PARP deficiency. So now we're going back to the replication fork. See? It's only bigger now. And suppose I have defective base excision repair. There's a gap there that isn't going to get filled very quickly because I've got a PARP deficient mouse, for example. Look at this replication fork. It's moving to your right. It's headed right for there. Okay? So these are railroad tracks. And one of the two tracks has a gap. And this is the train. This is a big machine here, which is like a locomotive, going to the right very quickly. Okay, so what happens? The migration of the replication fork converts the single-strand break into a double-strand break. See? Now, if this break is here, now if I start and do lagging strand synthesis, I can't copy this thing. Right? There's going to be a gap. Polymerase can't copy across the gap. But since this is a new replication, this is newly synthesized DNA, this is exactly the scenario for which homologous recombination was designed. Because you have a double-strand break here, you can do homologous recombination on the other strand, all right, to repair this break. Remember homologous recombination where we had the D loop, and then we copied the missing material, and then we restored the DNA, and then we're fine. So that's why the PARP deficient mouse was perfectly fine. PARP deficient mouse was okay, not because PARP was not important, but because PARP is so important, there's a backup system. Stupid me. I should have realized, right? How could such a cool enzyme that was doing such cool things not be important? It was because it was so important. Are you okay with me on this? This is pretty hard. Okay. I'm going to have to give you a break soon. So homologous recombination repairs the double-strand break. Now here's the kicker. This is why you want to learn about this. People have developed drugs that inhibit PARP1. And it turns out they kill BRCA1 and BRCA2 breast cancer cells. July 9th, 2009. Whoa. Now, you can imagine PARP1 inhibitors. How about PARP1 inhibitors? Now, BRCA1 and BRCA2 breast cancers only form 5% of all breast cancers. What about the other 95%? What if I treated them with PARP1 inhibitors? What do you think would happen? I don't hear. Nothing. Remember the PARP1 deficient mouse? Perfectly happy because he had homologous recombination. Actually, she had homologous recombination. So, the other breast cancers, this isn't going to help, right? But for the 5% of breast cancers that have homologous recombination knocked out, this is going to be really great. Will this drug have side effects? Well, it could, but it works only when BRCA1 is knocked out completely. Both alleles, both copies of BRCA1 are knocked out. Then you don't have homologous recombination. What if a cell has one copy knocked out and one copy good? That cell still does homologous recombination. So you've got a drug that knocks off the cancer cell where both copies are knocked out, but for a normal cell where one of the two copies is still good, it's not going to be very harsh. Yes?
So if somebody takes that drug, does it mean that there is no repair going on during the period of time when the drug is being taken?
So the question is, if somebody takes this drug, what's happening to that person's repair while the drug is being taken? And the answer is, if you took that drug, you would be back here, and all your normal cells would have little gaps in them, in their DNA, right? But not to worry. Your normal cells do homologous recombination, and your normal cells would repair that when the replication fork moves forward, right? And then it repairs that break. So, you're taking it, and you are shutting down base excision repair, but lucky for you, you've got homologous recombination. Even if you happen to be, I don't know what your genotype is, but even if you happen to be BRCA1 or BRCA2 mutant, you've got one normal copy, and that's enough to make homologous recombination go. So that's a great question. That's asking the question about side effects a different way. Uh-huh.
So that's a really cool basic science result. But
that's a really cool basic science result.
Right. So okay. So now you've got maybe presumably there was some PARP1 inhibitor that they used to do the study. But suppose somebody says, okay, well let's move down the road to make this into a treatment.
Yes.
What kind of time frame?
Oh,
10 years?
oh no, it's about to get approved by the FDA. This drug, let me show you. When something gets into here, then this is called the New England Journal of Medicine. This was a clinical trial where they treated women with breast cancer. This is coming very soon. I don't know if it's, does anybody know if it's FDA approved already? Huh? Not.
It's in phase 2 trials.
This was a phase 2 trial that was reported.
Yeah, triple negative, it was effective with triple negative breast cancer. And the company was just acquired for $500 million by a Swiss company.
That's cheap. Yeah, this is, so anyway, this is going to be approved very quickly because in the phase 2 trial, the women who got treated had hardly any side effects.
So how long ago then was the science understood? I mean, I'm trying to get.
Oh, how long ago did we know the science? How long did it take to get to this point?
Yes.
You know, I was ignoring PARP literature. I'm a DNA repair guy, and I was ignoring it. But, you know, there were people working on it. I think we knew about PARP and basic excision repair maybe 5 or 10 years ago. It's been kind of perking along for the last 10 years. Yeah. Yeah. But, you know, we didn't fully realize what was going on for a while. And some of this machinery was getting put together from different angles. People were studying homologous recombination. Other people were studying basic excision repair. So, and all of it came together. I think this is beautiful. And I think the reason I need to tell you this is you just saw how the 3 R's come together to make a drug that has no side effects, that can take these very unfortunate women with a terrible cancer and treat it all by, you know, that's pretty good.
So what company should we buy stock in?
I forget. I forget. I actually do forget. But you could look it up. You could look it up because these slides will be online, and all you have to do is do PARP 1, or you could do New England Journal of Medicine, July 9, 2009, and PARP 1, and Google it, and you'll get the article instantly. Okay? Yeah, it's too late to buy the stock, obviously. Okay, applications, recombinant DNA and new drugs. Do you want a 5-minute break? All right. Wait. No, I'm wearing a Foley catheter, so not really. No. But if somebody, okay, a lot of women were saying no, but you know, some of the men might want to go. If anybody, if you want to go, I won't be hurt. Where are the bathrooms? Up here to the right or left, just outside. So if you want to go, I won't be hurt. It'll be good. It'll be fine. I always give the medical students a break after 50 minutes. And then if one of my co-lecturers don't give a break, I go in and yell at them. Okay, so we're going to do applications, recombinant DNA and new drugs. Okay. So, history. I showed you Arthur Kornberg, 1957. 1973, the invention of recombinant DNA. And I'm showing you these characters because you might bump into one of them if you're on campus here. This is Stan Cohen, who has a lab just across Campus Drive in the main, right? 2 floors above Dean Pizzo. Okay, so he's still there. Stan Falco. He's emeritus now, but I see him on campus all the time. Herb Boyer. I don't see him because he was at UCSF, and Genentech's Herb Boyer. He left UCSF for Genentech, the boom in genetic engineering. Prince Charles picks a bride. So this gives you a time frame for where we were. And Paul Berg still comes to work every day. So you might bump into him. This is the Beckman Center right across the way. So these 3 guys with the red arrows had a midnight brainstorming session at a scientific meeting at around midnight. And they were hungry and they were looking for corned beef. So they found on Maui a New York-style deli, and they started talking about plasmid DNA. These are little bits of DNA that bacteria have in them that actually carry antibiotic resistance, for example. And they had a brainstorming session where they thought we could maybe use these plasmids, which are the bane of antibiotics, and manipulate them to do some good for ourselves, make them into a tool. So they started having ideas about that. Paul Berg, meanwhile, was working hard at Stanford, not on Maui, doing experiments with the SV40 virus, and in Building M, which is also, at the time, 1 floor above Dean Pizzo's office. And what happened was they came up with the idea of recombinant DNA. And so the outcome was Stan and Herb patented the idea. That turned out to be the biggest patent that Stanford and UCSF, I think, ever, the most lucrative patent they ever had. So Stanford and Stan and Herb also shared in the wealth. Paul won the Nobel Prize. And, you know, someone had to pay for the dinner on Maui. Stan paid for the dinner on Maui. So I'll tell you an interesting story. When I was first appointed to be a professor here, they had this idea that they would hook us up with a senior faculty member who could be our mentor. And I was really lucky. I got assigned Stan. And so Stan took me out to lunch. And I asked him if he would please pick up the check. I can't remember what happened, but it didn't work. So, anyway, he actually, in many ways, deserves one of those, independently of. And so the Lasker Award was actually recently awarded to him. And the Lasker Award is designed to try to make up for oversights that Stockholm had. But, you know, and Stockholm can always make up for the problem, because Stan has done so many other things that they may choose to recognize him anyway for other things that he's done. Anyway, so that's that story. The other part of the story is these are congressional hearings because people feared what recombinant DNA could do. They feared that some bioterrorist could create an organism that could wipe out the world. And so what ended up happening was Paul was the leader in recombinant DNA. And Paul decided that it was be a politically good thing if scientists put a halt on their own research before Congress got into the act. Because one thing you don't want Congress to do is to try to figure out how to regulate something. So Paul called a conference at Asilomar down the coast near Monterey, and all of the leaders in this field decided that they would have a self-imposed moratorium until they could figure out what would be safe. Okay? And then Congress didn't have to act because everything came to a halt. And since Paul was ahead of the game, the fact that he was stopping meant that everybody else felt that they had to stop too. And so we actually emerged from that with the ability to make recombinant DNA molecules, and you've never heard of an accident and so on and so forth. So things have come out okay. So in 1973, invention of recombinant DNA. And what you do is you open a plasmid, which is a circular piece of DNA, and you take into it, and you put into it, insert into it a foreign DNA molecule, say for something you want to make, say growth hormone, which is one of the first things that Genentech wanted to make. And what you do is you can make now this DNA by polymerase chain reaction, which is basically replication in a test tube. And then you introduce the recombinant DNA into cells. And the construction of this recombinant DNA is just DNA recombination in a test tube, right? So I taught you all about that, so you could do this. So now you could make some of your favorite protein, which could be a drug. Now, you might be interested in growth hormone if you have a child who's very small. But you can also make some very important growth factors. One is called EPO, or erythropoietin. And it's a growth factor for red blood cell precursors. And it stimulates the production of red cells. And the way it does that is I'm showing you a molecule of EPO in red here. And it binds to what's called the EPO receptor on the surface of cells in the bone marrow. And these cells in the bone marrow put together this receptor as a dimer. So there are two subunits that are the same as each other. And EPO binds in this pocket here. And the cell gets the signal, aha, it's time for me to divide. That's a red cell precursor. And it starts dividing and making more young red cells to go out into the circulation to help us breathe. Okay? Very neat. Now, the reason this is important is EPO is made by our kidneys. And so who are the people who are going to get anemic? Who do you think? People who have cancer, I heard. But since it's made in the kidneys, who else? Dialysis patients. Or anybody with kidney disease whose kidneys aren't working. Exactly. And so the good of this is it treats the anemia associated with either renal failure, patients on dialysis, or with cancer chemotherapy. And it replaces transfusion, which often takes a few hours, with a single intramuscular injection. Boom, it's in there. Okay? The problem, of course, is it takes 2 weeks to work for the red cells to emerge out of the bone marrow. But it's very convenient and very quick instead of a transfusion. It conserves the supply of donated blood. And this is a really big problem, because in the Bay Area especially, there's a shortage of blood. But the Bay Area imports blood from the rest of the country. So we get blood from the rest of the country to make up for our shortage. And it eliminates the risk of blood-borne diseases. So we do have a shortage of donated blood, especially here. In the Midwest, it isn't as bad. They seem to have different values there. It isn't because of HIV, by the way. The incidence of HIV in the Bay Area, of course, you know, is only going to be a few %, right? So it's mostly because people don't have time to donate. So my wife and I donated 3 of the 10 units of blood the blood bank, Stanford Blood Bank, collected this weekend. So the guys here, you could actually donate 2 units, which I just did, which is kind of fun. You can go swimming and see what happens, and things like that. So that's an advertisement for donating blood if you think you're eligible, because there really is a shortage. It's very sad to go in there. Because when I offered 2 units, they were getting close to closing, but they said, "We'll take you." and I discovered it's because up to that point, they had only collected 7 units. And it was the end of the day. So that was it. So the bad is that picture of EPO that I showed you is part of this advertisement. That might be Optimus Prime, I don't know. So, and EPO is really good for the Tour de France and other athletic pursuits. And the reason it's good is it is a recombinant human EPO. It has precisely the same protein amino acid sequence as human EPO. It's very hard to detect. It's a mimic of the human protein. So you can actually give yourself a shot of EPO and raise your hematocrit so that you now can carry oxygen much better to your muscles and become a better cyclist. And get away with it. So that's the bad and the ugly of it. Okay. Now I'm going to tell you some more things that are even uglier, because this is really ugly as far as I'm concerned. This is one of the most shameful things on the planet right now. It's just as shameful as people starving in other parts of the world, because this is a country that's actually wealthy enough to take care of its own citizens. So the view from below, the clinic, I'm going to give you 2 examples of how spending more money can lead to a worse outcome. Because one of the things you've heard about is, how are we going to insure all the uninsured? How can we afford to do it? And I'm going to show you 2 examples that amount to billions of dollars, where we ended up spending billions of dollars to get worse outcomes. And so this gives me hope. I mean, this was shameful that we did it, but it gives me great hope that if we structure the health care reform properly, now we're getting into things I think you're really interested in, we will be able to give better health care to everybody for less money. How much less? About 2-fold less. I'll show you why. Okay, so from the ground, pharma educates patients. This is an ad that they give to patients. Are you ready to start chemotherapy? Low white cell blood counts. How Neulasta? Neulasta is another growth factor, just like EPO, and it stimulates the production of white cells. Help protect against infections. Questions to ask your doctor. The question is, hey doctor, can I have Neulasta? Okay? The reason they're advertising is because it costs $2,500 for each injection. So the company makes a lot of money every time we prescribe this. That's a lot of money. That's the administration fee. Now, I'm being a little unfair. That's the administration fee if you don't have insurance. If you do have insurance like Blue Cross, they've negotiated the price downward to $6,500. It's a lot of money. So the hospital loves us oncologists. Medicare actually, Medicare only pays a little bit more than that. So doctors don't like Medicare. That's why the AMA is so scared that a public option would be just like Medicare. In fact, I think the public option just died today. Okay. So, yeah, so it may come back. They're going to give it another try. I don't know. Anyway, I don't really care what option they have as long as they design it properly. I'm deathly afraid they won't, because too many of the wrong people are lining up behind this with too much enthusiasm, and they have a lot of money to make. I wish they were a little bit more, you know, some of the big industries are really love what's happening now. So this is Aranest, a long-acting erythropoietin. Same company, same kind of advertising. Amgen makes Epogen and Aranest. Johnson & Johnson makes Procrit and Eprix. They divided the market. Amgen does the cancer patients. Johnson & Johnson does the dialysis patients. 2004 sales of EPO, $8.6 billion. Now, Amgen stock, this isn't a real graph. It goes from 70 to 50. It doesn't go to 0. But the price in US dollars in January was here, and then in the course of a couple months, dropped precipitously. Something happened. What happened? What's called the DeHanka 10 trial, November 2006, just before the stock drop. It was prematurely terminated. What was it? It was to determine if Aranest improves the outcome in head and neck cancer patients who are treated with radiotherapy. The reason for doing this trial was there were theoretical reasons for thinking that it might help, because cancer cells, when they're hypoxic, when they don't get enough oxygen, are actually resistant to radiotherapy. They don't die as easily. So it had a good theoretical idea. If the cancer cells get enough oxygen, they'll die more readily from radiotherapy, right? So the reason the company was really interested is there was a good theoretical idea, but what they were going to do is they're going to treat patients with hemoglobins less than 14. Now, 14 is a really high number, so they were going to get the hematocrit up there to where the cyclists want to be. If you go to the blood bank, they'll take you if your hemoglobin is 12.5 or above, and they'll take blood out of you. So 14 is really high. Okay? The result, 5-year disease-free survival, was worse on Aranest than the control. Whoa! P equal 0.02. Journal of Clinical Oncology, 15S supplement. That was a supplement because it's only an abstract. It hasn't been published yet, but it was reported at the American Society for Clinical Oncology meeting last June. Let me show you. Now, that was last June. So 2006, okay? The report came out last June, 2009. Look at this date, 2003. They were selling EPO since, you know, right along, 2003, 4, 5, 6, 7, 8, 9, all the way right through. This is a paper published in Lancet, not an invisible journal at all. It is up there with the New England Journal of Medicine in prestige. This is a Kaplan-Meier survival curve. This is months, time in months. This, on this axis, is what's called progression-free survival. At the beginning of the trial, 100% of the patients have not progressed yet, by definition. But as time goes on, more and more patients the cancer starts to progress. So as the curve goes downward, that means you're losing more and more patients because the cancer has progressed. So the lower the curve, the worse you are, right? So as time goes on, more and more patients get picked off by the cancer. Now there are two curves here. One is a placebo control, where you just give sort of normal saline. Boom, you just inject normal saline. Those patients aren't doing great. Head and neck cancer is a very bad cancer. So after 24 months, actually, only 40% of them are still cancer-free. But you gave some patients EPO, and look where that is. The curve is below. Costs $13,000 a shot. With insurance, Blue Cross, maybe $6,500. There's a lot of money. $8.6 billion going into this. I think we oncologists were spending more than the dialysis doctors. What's going on there? Well, I told you. There is this receptor called the EPO receptor that, when it's stimulated, causes the red cell precursors to grow. Now, when you name something, you usually name it because that's how you discovered it. It's called the EPO receptor because it was found on red cells. There was no law that said cancer cells couldn't have EPO receptors. And indeed, cancer cells do have EPO receptors. They have two kinds. They have the normal EPO receptor on the surface of red cells, and they have a heterodimeric EPO receptor as well. Not all cancer cells have this, but many of them do. And those cancer cells that have these receptors will respond to EPO by growing. So, that's what that diagram means. And we were doing this for years, even after we knew about this. It's disgraceful. $8.6 billion a year.
So was that study done, was that report done pursuant to a study on head and neck cancer?
Yeah, this is head and neck cancer.
So the company reported to the FDA?
This was published in Lancet. It was reported to the world.
Right, but did the company have an obligation to report that?
Well, the FDA knew about it.
Is the indication for the drug head and neck cancer?
Yes.
Or that is
Yes.
primary indication?
One of the indications for this drug is anemia of chemotherapy. Was that your question? Yeah.
But the drug wasn't being administered.
It was being administered to head and neck cancer patients. It was. How did it happen? Okay, write your congressman. We have a dysfunctional FDA is the answer. The question was, how did that happen? The answer is, we have a dysfunctional FDA. That's my opinion.
Well, how does this stuff kind of propagate out into oncology? I mean, so if something comes out in the Lancet, does everybody sit down and read this, if you're a practicing oncologist, or is
I haven't been giving EPO for years. So let me tell you why some people were giving EPO. The company, was engaged in a scientific discussion. Maybe this result is real, maybe it isn't real. Maybe there are reasons for this result. Maybe there are shortcomings in the study. Maybe, right? But let me tell you, a lot of people in private practice were giving EPO even when it probably wasn't the best thing to do, when the patient really wasn't that anemic. Why do you think they would do that? Well, the patient was tired, was getting tired, and chemotherapy is tough, and they said, well, maybe I'll give you some EPO because maybe it'll help you feel better, and oh, by the way. So there's a huge conflict of interest among physicians. And so one of the problems with medical care here is our system is based on fee-for-service. And this is how fee-for-service can be a problem. But I'm preaching now. Let's face it, I'm editorializing. Yeah.
How would the public option fix that?
How would the public option fix that? Let's wait. Let's stick with questions until the end, because I want to give you enough time to ask questions. But we can talk about how would I fix the whole system, but that's philosophical. Yeah.
So if the drug costs $2,500 for a shot, and let's say Stanford charges $13,000, how much of that difference goes directly to the doctor?
Oh, to me, nothing. Because I happen to be on salary here.
But not salaried physicians.
Huh? I am a salaried physician. Oh, private practice. No, in private practice, you're running a private practice, so that goes to support your practice. Yeah. Oh, yeah. Yeah. We can talk about those things later. I'm going to give you a short history of Vioxx. You all heard about it. Fewer gastrointestinal side effects, published in the New England Journal of Medicine in 2000. Between 1999 and 2003, there were 8 trials with independent endpoint committees that showed an almost 4-fold increase in heart attack risk of Vioxx versus naproxen. It turns out there were 8 trials without independent endpoint committees, and it turned out Vioxx was actually protective. All of these trials were sponsored by Merck. But the independent endpoint committees can't have Merck employees on them. 2004, Vioxx was withdrawn. If you want to look it up, Kerr in New England Journal of Medicine, 2007. The Victor trial was to see if Vioxx improved outcome in colon cancer patients because it turns out that there was some data that suggested Vioxx would prevent colon cancer because aspirin does. And aspirin probably does so through the receptor that Vioxx works on, something called the COX-2 receptor. Okay? So this trial had a good scientific basis to it, but the problem was that in the background, these heart attacks were going on. So I'm going to run real fast through this, but there's Vioxx biology that actually predicted the problem. There's the COX-2 enzyme here that actually stimulates inflammation. This is what you want to stop. Okay, but in addition to that, there's an arm of this pathway which goes through the COX-1 enzyme that does very important things for platelets to make blood clots and protects the stomach, okay, from ulcers. Aspirin and naproxen block both pathways. So what you do is you get relief from arthritis, but you also have stomach problems. So what Merck did was they had the best drug for knocking out COX-2 and leaving COX-1 alone so you could protect the stomach. That paper in the New England Journal of Medicine in the year 2000 was trumpeting their success in protecting the stomach. It was wonderful. And in fact, I think Vioxx, if we only had it now, we really ought to have it. Because there are people with arthritis who simply can't take anything like this because they have too many stomach problems. But the problem is, this was a very profitable drug, and they marketed it to everybody, even people without stomach problems. They also found out that this arm of the pathway stimulates growth of polyps in the colon. Therefore, if you knocked it out, you could prevent colon cancer from recurring, for example, or maybe even have it as a preventive. So if it were a colon cancer preventive, everybody over 50 would be taking it. Who knows how much money that would be? So that's why they liked that trial. It really had an upside. But the problem is, it could also affect this arm of the pathway. And remember, it could end up disrupting the clotting mechanism so that you could have unwanted clots in the blood vessels of the heart. And this is something they did know about. And when they designed the first trial that was published in the New England Journal of 2000, they specifically excluded people with a risk for heart attack. But that's okay, as long as they market it to people without a risk for heart attack. But that's not how it happened. So the number of people who died in Vietnam was 58,000, just to give you a rough idea. So has pharma biased the literature? Yeah. 26% of all the papers in the New England Journal of Medicine are written by ghostwriters. Somebody not listed as an author wrote the paper. And I can tell you that if a scientist has an article in the New England Journal of Medicine, they can't afford to hire a ghostwriter. It's probably, and I know that almost all of the pharmaceutical industry-sponsored papers are written by ghostwriters. I've talked to a ghostwriter about this. Randomized myeloma trials. This is a fatal disease. And when you do a randomized trial, one arm gets the best possible treatment that's known today, and the other arm gets the experimental treatment. Normally, you would expect experiments like that, if you really don't understand, if you don't know what the outcome is, you would expect them to favor the new treatment about half the time. When these trials, myeloma trials, were not sponsored by industry, it came out in favor of the new drug about what you would expect, about half the time. When they were sponsored by industry, they came out in favor of the new industry drug 3/4 of the time. So I think the literature is biased. What about oncologists? Where do they fit? This was our convention center, Orange County Convention Center in Orlando. Stop by the booth, 3324. And down here, this is what I got in my email from Cam Stark. I think those are $100 bills. Benjamin Franklin, who's he? 50? 100. Fistful. They send that to us. And I responded. I took it and put it in my talk, but, other... high drug prices yield high administration fees for oncologists. So these are, pleased to announce, a practice enhancement service line. Practice enhancement. Well, in case you don't know, former editors of the New England Journal of Medicine are really angry. The truth about the drug companies on the take. It's easy to see why they have books like that.
Why do high drug fees lead to high administration fees?
Why do they have to? The question is, how do we get away with charging these high administration fees? These things are just a shot. How difficult is that? Boom, right in the arm. I don't know. Why do the insurance companies decide how much to pay? It's complicated. But, it's got to change. It has to change. I think it really needs to change. So the view from above, I'm going to go through real fast. This is the whole outcome. This is California. This is a graph of annual Medicare spending per beneficiary. This is overall quality ranking. This is where we are. You can see the line actually gets worse the more you spend. Now, on the one hand, that's disgusting. But on the other hand, you can understand why I'm so optimistic. Because if we spend less, we'll get better outcomes. Okay, this is healthcare spending in the U.S. as a fraction of GDP. So the GDP is growing and we are spending more, but we're outpacing GDP. Back in 65, we were 6% of GDP. We are now about 17 or 18% because the rest of the economy collapsed, and we're headed towards 20%. Well, it can only go so far. Okay, this is a graph. The richer a country is, the more it spends on healthcare. That makes sense. We want our citizens to be healthier. But, so we've got Turkey, Poland, Mexico down here. GDP per capita. This is healthcare expenditure per capita. There's the U.S. Even when accounting for the fact that we're one of the richest countries, we are about 2x over the line. What about quality of care? This is a double graph. The red dots are per capita spending. This is where we are in terms of spending. The red dots and all these other... Now, these countries include Japan, Sweden, but they also include, look at this, Costa Rica, Cuba, Chile. Look who's down there with Costa Rica and Cuba. Tied in life expectancy. In WHO rankings, I think we're right there with Costa Rica and Cuba as well. In WHO rankings of quality of care, not just in average life expectancy. So how can we fix the cost problem? This is the pie. Drug costs are rising fastest of all, but physicians earn money for treatment, not prevention. And hospitals succeed when patients fill their beds. So I have mixed feelings about expanding the hospital because I think the ideal system, we should be hoping that the hospital shrinks. So that used to say 5 cents, but anyway. For 5 cents, yeah. I think I went on a little too long, but we...
So you expect it to stay at the molecular level, but we've gone way up to dealing with the healthcare system, and I think, as Gil knows, we share many of the same views about this, and I think this is an extraordinary time that we're facing today for all the reasons that were articulated. So because we still have just a couple of minutes, let's see if we have any questions that relate to any of the topics that have come up so far.
On the website, we've had documents from this organization and that organization, which is the topic. I was wondering whether it would be on the website the
The question is, can you access the shots that I found in my presentation on the web? Yeah, I found those all on the web, and I'm going to send Kathy my PowerPoint presentation so that if you want to look at them, you can. And also, you should be able to find them by doing a Google search.
So we're gonna be posting.
Yeah, I'm going to post a whole slideshow on the web.
Offer now and subsequent presentations. We'll be posting them online. Okay. Yeah.
So talking about the repair R, you were talking about before.
Yes.
Specifically with a virus that infects the cell and changes the DNA.
Oh, when the retrovirus infects the cell and integrates itself into our DNA.
Correct. So now you have millions of these cells affected. How does the body repair that without killing the cell? Or can it?
Sorry, that cell is done for. Yeah, because what happened is the question was, what happens when a retrovirus invades one of our lymphocytes, that part of our immune system? That's what HIV does. It invades our immune cells. That cell now has a copy of the retrovirus in its genome. Well, it's only going to recognize that DNA. That cell is going to look at that DNA and say, that's mine. So the retrovirus is so clever.
So let me just tie this back for a second, because I know we're going to be running out of time shortly. So I think that what you've heard from Dr. Gill is really the extraordinary story of not only how DNA works, but how this whole process of recombination takes place. And I think the illustration that you gave about how that in some ways relate to the whole recombinant DNA technology was an extraordinary one. And that actually is what spawned most of biotechnology here in Silicon Valley and elsewhere. But he also told the other side of that story, which was great discoveries can also be greatly abused, and it really does depend upon whether or not we match utilization to need. And sadly, that is where our system is really out of whack. So we're a nation that has, by far, the greatest bioscience discovery engine in the world today. And as you've heard, and I stated last week and will continue to reiterate in other settings, we spend more on healthcare than any other nation in the world, with nothing to show in the population basis other than that we're number one in administrative overhead. And you've heard some of the reasons for that tonight. So I think we've had a rich, exciting discussion tonight. I hope you've enjoyed it. Next week, I will actually be missing because I'm giving testimony to a congressional committee on healthcare reform. I've been back to Washington many times over the last several months. It's hard to know where things are heading as it stands, but I think with the Senate Finance Committee's decision today, I doubt they'll want to hear from me next week, but we'll do our best. But next week will be an opportunity for regeneration, and Jill Helms will be speaking about stem cells and regeneration, so please come back, and we'll look forward to your comments and questions. Thank you for being here tonight.
For more, please visit us at stanford.edu.